Hey, good morning, everyone, and welcome to UroNurse. Don't forget to like and subscribe. Today, I'm diving into a story that sounds like science fiction, but it's very real. Imagine bacteria that can literally eat tumors from the inside out. Yes, you heard that right. Researchers are experimenting with Clostridium sporogens, a soil-dwelling bacterium that thrives without oxygen. And, as it turns out, the inside of a solid tumor is the perfect environment. Low oxygen, plenty of nutrients for these microbes to survive and grow. Chemical engineering professor Dr. Mark Aucoin explains it simply. Bacteria spores enter the tumor, find an environment rich in nutrients and low in oxygen, and just start eating these nutrients and growing in size. In other words, the bacteria are colonizing the tumor from within, essentially ridding the body of the tumor. It's kind of like a little Pac-Man if Pac-Man lived inside your cells. You know, that little thing, chomp, chomp, chomp. But here's the catch. These bacteria normally cannot survive on oxygen. So what happens is they reach the surface of the tumor. Well, there's the kick. They encounter oxygen-rich environment of our body. This is where the genetic engineering comes to play. Scientists added a gene from Clostridium aminovalsicum, which can tolerate oxygen. This allows the tumor-eating bacteria to survive longer as they approach the tumor edges. But safety is critical. If these bacteria could survive anywhere in the body, we could fix the tumor but create a whole new problem. So researchers used a clever trick called the quorum sensing. So you're probably asking, what's that? Quorum sensing is how bacteria talk to each other, essentially counting how many of them are in a colony and adjusting their behavior accordingly. The team engineered C. sporogens with quorum sensing machinery from Staphylococcus aureus and used green fluorescent protein as a marker to track success. Think of it like a biological circuit. Instead of wires, they use pieces of DNA. And the goal? To turn oxygen resistance on and off, depending on whether the tumor has been completely eaten. Senior author Dr. Brian Ingalls calls it Hungry Hungry Hippos, the synthetic biology edition. Got a lot of kids games here. All right. This research published in the ACS Synthetic Biology represents a major step towards safe targeted bacterial cancer therapy. While we're still in the experimental phase, the hope is that one day tumors could be treated from the inside out with minimal impact on the rest of the body. So the next time you think of bacteria, don't just think of germs. Think of them as potential cancer-fighting allies. That's it for today's Spotlight from UroNurse News. Make sure again to subscribe and like our channel and tune in tomorrow for more. Hey, speaking of tomorrow, ever wonder how clinical trials went from fake exorcism to life-saving modern medicine? Tomorrow on UroNurse News, I'll take you through the strange, shocking, and sometimes unbelievable history of clinical trials and how those lessons shape the research we rely on today. So be sure to tune in tomorrow and check it out.
News, clinical guidelines, and research insights delivered weekdays at 9 a.m.
UroNurse, where the experts meet. Online learning, practical and sweet. Every Saturday, 9 a.m. Central Time.